All right, so it looks like we have already got some people lining up at our microphone, so we'll take in-person questions first. I'll start left to right. So go ahead with your question.
Okay, I'll go start. And thank you for all the speakers this morning. Alan, again, thank you so much. You gave us a really wonderful view about the history, at least on the topic, and I really appreciate you being here and sharing your perspective with all of us. And I do have quite a few questions, but I want to just ask one for now, and I'll give others some chance. And this question is related to Jörn's presentation that you described this challenge for the suspension material that has excipient in the formulation, in the drug product, that created a challenge during the measurement. So I want to again reiterate what you just mentioned in your talk, which is this challenge is very practical and it's present in not just one product, it's many products. Because we're not just measuring the suspension of particle of a pure component, but we're measuring a mixture of component and excipient, which are commonly polymeric material, and they do interfere with the analysis by laser diffraction, which bring the, what the two approaches that you mentioned. One approach, which is we published the paper, hopefully people more reading about it. There are two papers about these approaches. One is just from background subtraction, and the other one more measuring the product. But I do like what you are making the comparison between this approach one, which is based on the placebo approach, which is not blessed by any of the vendors sitting today in this workshop. And the method two, which is I think we see more often used by the applicant, which is just truncate or in some way statistically or mathematically the data that's presented with the justification that what we care about is this range, and this range we have some data to show that we don't need to take a look. But I do, maybe a little bit biased, I do see the risk of approach two, from a regulatory point of view, it's a little bit higher than the approach one. Although this is not my question. My question is for all the vendors that you're here, especially if you have laser diffraction instrument, can you look at the paper and critique what could be the potential limitations of changing the data basically at the detector level by changing the or using the background that come from the placebo, which has all the excipient in them, essentially to remove them before analyzing the drug product, which I certainly acknowledge there are still risks. But I'm interested to hear, at a minimum, if you can consider looking into that and maybe can come up with some instructions or white papers about how to practically address this particular challenge. And even if means what we have published is incorrect, there is a better way to do it. I would be really happy to hear what would be the recommendation, because this is a practical problem that we are facing. Jörn, sorry, I used your presentation to asking this question, but this is not really directing to you, but more for the maybe others on the podium and also the audience. Thank you.
So I guess the short answer is, your question is, would we look at it? I certainly will. Only about 87 pieces of scrap paper, but it should be interesting because you're really subtracting before and after transform, and what sort of trouble that gets you into is not yet clear, but the paper might make it clear.
And maybe just a short comment coming from the industry. So I really appreciate the comment from Xiaoming. I think it's really on the point. Yeah, it is also our duty as industry. There are different ways how we can look at the same problem and also how we can solve the same problem. Really, the purpose today was maybe to show two aspects. As I said, I hope maybe some of you can also share with us the third, fourth, fifth option. It would be great to hear, to talk either now or during the discussion. But what still, let's say, gives me confidence is that in this particular case, we need to consider that it is always a product specific. It's not a one universal solution. Still, using both approaches, we came to the same conclusion, meaning also that in a way, looking from two different angles, we came to the same. So this gives me, let's say, a confidence that in this case, either both approaches are wrong or both are giving the same result. Thank you.
I may add that at the end of the day, with 2 and more materials in the sample, you must have specificity in your validation. You must show that you can differentiate between the particles. If they do not overlap, that's an easy case. And usually they do. And when they overlap, you know, what we have succeeded so far is only including with microscopy or shape analysis. We have succeeded in one case to differentiate the drug substance more than 95% using shape analysis. And in other cases, we use Raman spectroscopy either to validate the method to show the specificity or to use mapping where we can identify the specific particles, but not in laser diffraction.
I can comment on your study. I mean, it was very distinctly 2 separate peaks. And in that case, if I were to approach as a vendor and customer came to me, I'd be like, okay, you have a clearly identify your IPI, you have clearly identified placebo, measure them separately together and then see how the data looks, I mean, which you did. I don't know how FDA will approach that, but as a vendor, I have recommended, okay, measure 3 different times, one with API, one with placebo, one with together. And as you can clearly distinguish 2 separate populations, if you do, I think your data is very valid in that sense. That's what I would say as a vendor.
Okay, any other input on that one? No? Good. All right, let's roll to the right-hand side here.
Okay, cool. So I also have a bunch of questions, but I'll save it for one and then hopefully I'll, jump. Oh, wow, it's grown. Yeah, well, we'll see. So before I ask my question, just I wanted to have a shout out. So Steve, Yusuf, and I were talking extensively about working groups yesterday. I completely echo the need for it. So there's multiple avenues that we can possibly do this. One of them is an IQ working group that Merck is already a member of. So for those that are interested of such a working group idea, getting together, publishing white papers and things, please email me so that I can compile at least a list and, get the conversation going. Okay, that's not my question. My actual question is the following. So if you look at excipient particle size, so using an API, you process it, typically monomodal distribution most of the time, but excipients are often bimodal distribution, right? And it's real, it's really there. It's not a ghost peak, it's consistently there. It's just the way that these things are sometimes commercialized and made by manufacturers. If you have to develop a particle size method on the excipient, which is fundamentally a bimodal distribution, what you see sometimes is that D50 is meaningless, right? Because it fits right in between the 2 valleys. So the question, open question to everyone here is, what approach should we take in method development and also in validation? Like, should we, for instance, change the D10, D50, D9, do something else so that we're tracking sort of the peak of the 2 distributions? And if so, what validation acceptance criteria do you apply for repeatability? Because if you look at USP 429, it's based on the repeatability, 15%, 10%, 15% respectively for D10, D50, D90 when the particles are larger than 10 microns. So any thoughts?
Yeah, a different specification would be, I think would be acceptable as long as you justify it. But what we usually do is when the D50, the approach to give D10 and D90 more than D50 in the specification is because they're sitting in unstable part of the distribution. And in this case, what we do also for the D50, we double the specification. So we widen the specification for the D50 because it's sitting also in unstable part. This is what we usually do. We justify it and we submit and usually acceptable. But to set a different specification, we did not try that yet. But I think it can be accepted and, if it's justified.
Yeah, because it runs the slight danger of arbitrariness, right? Because you can get really creative with this stuff. You can go like D19. You can go D21. And then, what is the acceptance criteria between those two things? Should those change? But what it seems like from your perspective is you've kept the three metrics, D10, D50, D90, and just emphasized the edges more, and hopefully they sort of catch whatever.
There were
variability.
cases where the same excipient was previously tested with sieves, and when we move to laser diffraction, we still keep the sieve specification, which is not D10, D50, D90, but this is the rare cases.
Yeah.
Just a recommendation, maybe still recording the D50, and rather than controlling off a median, maybe a mode size. If you notice those peaks in a consistent position, you could go with the peak position and peak width as additional information, and still keep your edges, your 10th and 90th. Or use the earth mover distance.
Can I actually ask a question? When you do the bimodal, whatever the distribution you get, do you see the different peak size each time, or are you getting consistent data there?
So, within the overall process, there are variabilities. So if you're at a certain edge of the process accepted range, one of the modes is larger than the other. And then if you're on the opposite end, even though you're still within the process accepted range, those shift. Yeah. And it's just the way that the excipient is. Yeah.
Could it maybe be a case where you still do the 10, 50, 90 as traditional, and then add something extra kind of on top to better characterize what's going on, right? Like a result above or below a certain size, or a result in certain ranges, and use those as additional criteria while you still kind of keep the 10, 50, and 90?
Yeah, so, that's one of the alternative strategies, right? It's like percent of particles less than X size, greater than X size. And
Right.
then those are determined by where you are in the mode. But should those, supplement the D10, D50, D90s or supplant them?
I would think supplement, not supplant.
Yeah.
I would also add that it also specific to that specific excipient function. If it's, the functionality of this excipient will also, if you're looking mainly for the smaller particle of this excipient, so it'd be justifiable to add more specification to the lower part of the distribution.
Yeah.
Great, thank you.
Thank you.
I guess we'll go over here.
I have maybe a naive question for Dr. Allen. So when you discuss the reproducibility, you said add another sample, but then you also said add another batch. So my question here is, I think the whole 3 R's to validate the method is to build confidence in the method. So here, when you say add another sample or another batch, what if the second batch is actually variable to the previous batch, or is like for the 3 R's validation, does it make more sense to maybe stay with one batch?
Well, you say, what if the second batch is a variable? It is, period. So the second batch is different from the first. It's whether those differences affect the product performance in any way. And that's what you've got to look at, those particular differences. But that's the effect of what people don't often consider, is that the sample is a variable, and the instrument is very good at picking up that variability within the samples that are presented to it, and very often small amounts. But that's there's only one way of looking at the same sample time and time again, and that's recirculating it wet. You can see those changes, but anything else is a change to the sample, or change to the operator, or those variabilities. So those all have got to be examined, but that major factor is the heterogeneity of the sample. And I use that quote very often, heterogeneity is the bane of particle size analysis. Tails of distributions are pigs to work on and analyze. I have more questions than answers.
Yeah, I'll take a step back to, elaborate a bit more why I asked this question is from generic development, right? One is RP, like we have seen efficiency issue, for instance, simply because the method validation didn't use the RLD, and that thinking has been updated. Now, if the method validation even is based on test product, as long as the overall validation is complete, then we would not issue deficiency simply because RLD is not used. Then another related question is our PSG generally recommend 3 lots for the benefit to know actually the potential batch-to-batch variability for the RLD. But then we get frequent question, there's only 1 lot or 2 lots available on the market. What can we do? So in general, if the applicant has evidence to support the availability is an issue for not being able to fulfill the 3 lots, that's acceptable as long as they test the 3 lots of their tests. So that's why I was wondering, so there's this always concern or debate when we say validation has to be on RLD is what if there is batch-to-batch variability? So that's why I asked you that question.
Yeah, sure. You have to do your error variability, your error budget. You have to look at that, what those variabilities are. But more importantly, you have to ask yourself, does that make a difference to the product performance? If X changes by 10 microns, is it a problem? Is it not? And very often those studies are not done. I've asked those suppliers very often, if your size changes from 10 to 15 microns, your D50, for example, what happens to the product? No idea. Somebody's just set this specification in thin air without any reference to what's happening. Is the dissolution affected? Is the product performance affected? That's the big picture. But thank you for the question.
Let's go over there.
Okay. Good morning, everyone. This is regarding the discretely appearing, randomly happening ghost peaks in the light scattering, laser diffraction analysis, especially with we use small one. And especially it's a nightmare for the QC scientists when they go for analysis. It randomly happens, either API analysis or suspensions, where we prove that there is no agglomerates in microscope, but still it's happening randomly. For example, in liquid method, wet dispersion method, there are 3 measurements, average of 3 measurements are reported. First 2 measurements will be okay. The 3rd measurement, suddenly one ghost peak appears. There is no valid, we tried to get the expert's opinion many times from the Malvern, and we couldn't get the, clear-cut one answer. Is there any way we can mitigate? And if it happens, how to scientifically address that? It's become an investigation, how to scientifically address. That's our question. For example, one more thing, I can give you background. I used for the past, the 20 years back, I used to use the Malvern Master Says S, and after it was 2000 and 3000. I don't remember anything happened with S actually, because those ghost peaks, I didn't see those times. In 2000 started and 3000 also happening actually. I don't know whether due to the instrument sensitivity, maybe you can address that.
Are you measuring in water when you're doing this?
Yes, yes.
It could be an instrument sensitivity issue.
Okay.
It is sensitive enough to pick up small kind of momentary peaks, if you will, right? If it passes through one time, the instrument is sensitive enough to grab that. So that could be why you're not seeing it every run, because it's not consistent throughout the sample. And it is just in the MV, right? It's 120 mils passing through. So it is possible that it's only every so often, if you will, and a longer measurement time might help work that out.
Okay.
Put a visualization unit on. You can do that with the Malvern, I understand. Look at what's going through the system and pick up the odd, maybe a bubble comes through or something else comes through. I don't know. And volatile solvents are typically a problem. Any fluctuation in, if you're measuring in hexane or heptane, things like that, acetone, they're all problematical because they cause refractive index variation in the solvent, which will cause the laser beam to steer. So I would always have a look at the sample that's going through the system. That's going to give you a lot of information.
Okay.
Okay, we've seen three different kinds of peaks like that. It's a thermal peak, what we call a thermal peak. And then when we keep the sample enough time next to the system in the same environment for enough time, that will reduce the phenomena. It doesn't disappear. We've seen bubbles. And in this case, either we continue to see it, we change the method completely. And we have seen peaks related to actual aggregates. So what we do is we take sample from the system, from the master we go to the microscope and start examining this. But what we do also for regulatory perspective, we have generated a guidance, internal guidance, where if it happens once, the researcher and the QA sign a form. It's not to open an investigation, but to sign the form. And then we test the sample again. We go back, examine the validation result if we have seen something like this. And then if everything is okay, we sign the form and we retest the method. If we see it again, we open an official investigation.
Okay, thank you.
Go over this way.
Just to talk to Powell's question a little bit. I think we heard this morning about these inline image analyzers that can be added to laser diffraction, and they're very helpful for picking up on these artifacts. I know did a demo once at a major US pharma with laser diffraction with one of these add-on image analyzers, and the customer said, I don't see how anybody could ever develop a method without one of these accessories. So it's really helpful for quickly troubleshooting these artifacts or head scratchers. But my question is, so laser diffraction instruments, now we see a lot of vendors that have a lower size limit spec of 20 nanometers or even 10 nanometers, which seems to suggest there's a very strong overlap between laser diffraction and DLS on the lower end. Not everybody's fortunate enough to have both instruments in their lab, a DLS and laser diffraction. When Anton Parr measured the samples provided for the workshop, we were fortunate to have both in our lab, and we ended up using both instruments and found that it was useful to have both. But maybe if a customer or a pharma company only has laser diffraction and they're going to measure a nanometer sized sample, whether it's 5 nanometers, 20, 30, 50, 100 nanometers, that's within the stated measurement size range of the laser diffraction instrument. My question to the panel is, what have the instrument vendors done to provide increased confidence in accuracy of the size measurements in, let's say, the nano range, sub 100 nanometer range using laser diffraction?
Well, good question, Joe. One thing I would say are there are very few particle size standards in the sub 100 nanometer region, and you might have to go to biology, to things like cholesterol or other things, to find materials that are there that can validate performance. And as you know, below 100 nanometers, you're in the monodisperse sphere category, from 20 nanometers up with the old Duke standards, the thermoscientific ones. So the verification of a system in that area is invariably with the sub 100 nanometer materials. But going back to ISO, you've got that stated range of 0.1 to 3000 microns as a working range. Below that, then I wouldn't say all bets are off, but one needs to take much more care with what you're doing down there. Tails of distributions down there, maybe if the entire population is below 100, I wouldn't be looking at laser diffraction. I'd be looking at DLS or SAX or some alternative method.
I would add that we never went that low with laser diffraction. Usually, you know, at the end of the day, you need to set the specification for D10, D50, and D90, and you need the distribution. And going below 400 nanometers with laser diffraction we've never done that before.
So so as Alan mentioned, first, there are standards available in that size range. You encounter them a lot with DLS. It's not a lot of fun to operate there with laser diffraction. And so often someone who has a single instrument is going to have some compromises, although this vendor will certainly help you get a DLS instrument for that size range. I'm sure everybody else will as well. Yeah, so you can validate in that range. Whether the validation is less expensive than getting the right instrument for the range is another question.
Below 100?
Nanometer?
I'm asking if below 100 nanometer?
Yeah, there are standards.
Yeah.
20, 30, 40, 50, 60. Yeah.
Thank you.
Okay. So on behalf of online attendees, I just want to ask the panelists following question, because many of you mentioned of those DLS and the laser diffraction, they based on like assumption of spherical basis. So some like online attendees actually want to like get some information about what's the impact of particle shape on the PSD, particle size distribution, and is there any threshold value for like circularity of the particles where the PSD analysis below that threshold is no longer like considered like accurate? So I think,
Again, I can comment on that. If your aspect ratio in old parlance of a particle is greater than 5:1, they'll tend to flow a line in the system. So they'll align like rods in a river. They'll go through here. They won't go through like that. And you're looking exactly the same system as you would under a microscope, where it's lying flat to the plane of the measurement. Below 3:1, typically the particles will tumble randomly, and you can show that you extract a correct volume from that. In the intermediate range, 3 to 5 to 1, who knows what happens? But they lined up in sticks like pneumatic liquid crystals or smectic liquid crystals. So there is a flow alignment effect to consider as well in odd-shaped particles. But odd-shaped particles still scatter light. Random particles still scatter light. You don't get that scenario. You can, in certain circumstances, move the particles, and you can see the end of a rod and the other aspect of a rod, and you can, from certain cylinders, see the bimodal distribution from one end and the other end. So, irregularity is always going to be an issue, which is why you need some form of imaging. That's why you need some sort of look at the particles in order to understand what's really happening.
Okay, thanks.
All right, let's go over to Christine.
All right. Hello. So I consider myself an end user, so I do measurements quite frequently. And so I always look at things like uncertainty in the reported values of any software. So it's great to see that there are several different vendors. It's not just one or two. But with that comes a lot of complexity with understanding how each company has their algorithm set and all the different tools and aspects that try to make it more user friendly. And I know there's been a lot of conversation here yesterday and today about, how do we become more transparent? How do we work together? And so my question is mainly for the instrument vendors. And that is, how do you balance adding on additional algorithms or tools in conjunction with making method development easier for the end user? So if you have things like filters, how do you anticipate an end user who may not have an extensive light scattering knowledge to know when to turn on and turn off that filter? So that's kind of the frame of mind.
I guess I can comment on that because I work for 3 different vendors and went through a couple model changes for all vendors pretty much. So even within the same vendor, when you go from old model to new, there's change in algorithm because, we're implementing new optical system, new detectors, new laser source. So the change is not like you just have to deal with it. But most of the time we'll always implement, or all the vendors will implement some kind of old algorithm function in this new software to what we call I call it kind of make it dumber to match the old data. But again, I think that's why the discussion yesterday was great, because everyone's having all these specifications that's set. And once they, may say the old instrument just kind of dies and they need a new one, it's obsolete and they cannot get that anymore. And they have to face the reality where, do we want to revalidate where we have to pull the product for a few months, or do we just go with the new software and not deal with it? Or do we just match the old specification and ask vendor to do that? And based on my experience, most of the time, I just have to match the old data, because the industry is just so resistant to adapting a new set of data, right? So again, I wish there was an easier solution where, every time we make an upgrade, it's always the same. But as a vendor, we are also trying to innovate and improve our products. And with that comes with some data change. So it's really hard to convince our customers per se to, okay, change your data, this is better, because obviously they have a different set of purpose and objectives. So yeah, we deal with that in real time, but I don't think we have a clear solution how to kind of make all this innovation at the same time, keep the same data. So
Sure.
yeah, that's the battle that we fight.
Yeah, I would say we have a similar approach, backwards compatibility with legacy algorithms. Some of them are decades old, but then maybe the bigger or more important answer is just offering good application training to, things like tooltips built into the software that might be able to lead you towards the right selection that prevent you from calling me or emailing anybody else on the panel.
Yeah, I think it's a combination of pragmatism and training. Right? We need to, as vendors, I think we owe everybody in this room a responsibility about being honest about the capabilities of things, right? Joe just asked a question about the lower size range that we all kind of collectively claim, right? And sometimes we get a little bit lost in the marketing weeds versus what the instrument can actually do. And I think when we're talking to an end user like yourself, it's important that we kind of have a pretty open and honest dialogue. I think it's also important that we provide you with the training necessary, right? So we can walk you through what is new on this new version of the instrument. Here's where you stand to benefit from it. Here's potential pitfalls. And then we can have the conversation about the regulation surrounding that space also, right? Because we're all very cognizant of the fact that sometimes you just have to match the spec and that's all that the end user cares about. But there are other ways to approach it and say, the predecessor instrument was as accurate and precise as it could be for when it came out. And now we've moved steps forward. And now this is as accurate and precise as it can be right now. And very kind of broadly and generalizing, I have found the agencies to be receptive to that, because everybody likes steps forward. Everybody likes progress. It was kind of the theme of some of the questions yesterday, right? How do we get better kind of collectively? And we all have to, I think, take a step forward together if we're going to get anywhere.
Thank you.
Yeah, I just wanted to add one thing. I'm not representing vendor, but my request is the vendors, I think before launching a new version or an upgrade, they need to, have an SMEs in the industry where they can contact and ask questions and be prepared for what the industry needs, because we're not aligned.
I agree with that. And I know that we do have some people that we are in touch with. I mean, obviously everybody would love to have the newest, best thing in their hands, right? But I think conversations are had, and maybe there was a conversation about working groups earlier. Maybe that would lead to more fruitful things for both sides.
I think before I take one more question, I just want to say that this is a great point for a secondary discussion, as far as there's obviously a sticking point between the necessity for revalidation, the vendors trying to help us, do more precise measurements, and, the industry actually accepting that in the auspice of possible regulatory hurdles. So this is a really great topic that we should definitely try to capture. So we can talk about this more later.
There's always a challenge between a better result and the same result. And that's
Absolutely. Let's go over here now. Thank you.
Okay, my second question has to do with treatment of reference standards. So when you get reference standards, you know, you say, some people, you do like a daily check, where before you use the instrument, you load, you know, latex or glass beads, right? You run that and make sure the instrument's good. And then after that, once it's good, you run your, you know, API or whatever with your method. The question is, vendors typically supply a particular method for a particular material to run, right? Sometimes we see the issue, and this is like largely like a quality versus R&D thing, where sometimes we get asked to demonstrate that my method, which I've tailored to my API, is suitable for the reference standard as well. You know, sample prep and everything. And this is like largely a quality thing, right? So I know what our vendors are gonna say, right? But I wanted to have, again, this open discussion of, you know, should that be a thing, right? Because if I'm pushed by, say, quality to do that and say, you know, with my particular method, I can't find a suitable reference standard for my particular dispersant, that sort of leads to a choke point that is, you know, from fundamental design flaw issue rather than anything else. So does my method with the same sample prep and everything have to run on the reference standard itself after you've demonstrated that an instrument's working?
No.
No.
You know, I'll expand a little bit more. Well, that's the total answer. When we're using reference materials to check instrument performance, we are looking at questions of whether the optical design is doing what it's supposed to do. When you have a polydispersed standard, you're looking at whether the pumping system is doing what it is supposed to do, and so on. We're not making any tests of chemistry, and there's no particular reason to believe that just because polystyrene latex is happy, anything else is going to perform chemically the same way. So kind of physical chemistry doesn't take a vacation. We can check the raw measurement with standards. But but there's nothing in our checks that invalidate the instrument for other liquids. And that may be what you need for your material.
Yeah, you're just verifying the response of the known material when you're doing that performance check, right? It has no bearing on your method working, not working, et cetera. It's just showing that the instrument can give the response that's expected given the known standard.
And the specification for the standards is much narrower than any drug substance, and that's why the method shouldn't be changed otherwise.
Yeah. So completely agree in all fronts. What we need sometimes, I found, is literally an end result, like a working document or something that we can provide to quality and say, no, we shouldn't do this. Right? We shouldn't do this. You're going to cause me problems. And sometimes it's as simple as, okay, where's your reference? For not the material, what I mean is like, show me something that's like accepted in the literature, something like that. So I feel like some of these sort of echo back to, I don't want to keep harping on the working group thing, but, you know, possible outcome as a result of the working group.
I think we kind of touched on it yesterday. I think it might have even been Billy's talk. It sounds like your quality group kind of has an eye towards more traditional analytical techniques, like they're looking for LC acceptance criteria on a diffraction measurement. That's never going to happen. It's not a thing that works. It's not a thing that makes sense.
Yeah.
Right. It's not applicable. It's not fit for purpose, if you will.
Yeah. Yeah. But then this isn't just unique to, you know, the particular quality group that we work with right now. Previously, I was at a generic company where I had that same thing for dynamic light scattering.
Yeah. I mean, I've definitely had that conversation quite a few times over my career with quality groups. So yeah, I know it's unfortunately kind of an industry-wide issue. I think groups like this are really important. Forums like this are really important, right? I think there's so many different analytical techniques. Everybody can't be an expert on all of them. So the quality folks, they're doing the best they can. They just don't know the question to ask. And maybe it's the responsibility of kind of all of us collectively to help steer them the proper direction.
So I can say that maybe a more immediate solution for you, we have service instructions for all of our standards. That could be the paperwork that you need. The alteration of the sample preparation is a probably the biggest problem I see, but assuming that they're similar, normally you just add directly to water, right? For most of these, it could be possible that you could take that result and do a recalculation, assume that it's in a different solvent or something like that. It's probably more like a half measure in that case, but maybe that's a way of jamming a square peg into a round hole for right now.
Time, we'll take these last two in person, and then if we have time for an online question, we'll do that.
So, good afternoon, everybody here. So, my question is about how do you handle hygroscopic substances? Tried doing it in a dry dispersion. I tried doing it in a wet dispersion. And the moment I try to, so I have a scenario that I have to develop a method and I have to validate the method. So the moment I take the sample out of the bottle or the container, it either use a dry dispersion, it gets stick to the instrument. I tried doing it in a wet dispersion. I experienced the same thing. I tried a wide range of solvents, starting from water to oils and everything. So what is your approach in handling hygroscopic substances? And is there any guidance from the FDA side?
Well, if your sample is sticking to parts of the instrument, it's telling you something about the sample. It could be an electrostatic attraction, it could be a moisture or hygroscopic attraction. So it's obviously eliminating that in some way and dealing with that. Maybe in a dry system, you need to make sure the system's properly grounded. In a wet system, maybe you need a surfactant or something. But it's important information to note if you've got the material sticking to other surfaces, you know, that is an important indicator of something that's going on. Used to be able to get radioactive ones that you could wave over dry samples and get rid of all of the static. I don't know. I don't think you can get those materials anymore. But yeah, it's telling you something. What it's telling you depends on the material and how it's being handled. It also tells me something about the health and safety considerations of that material as well. So I'd be worried about that in the laboratory as well, opening bottles and having material go around and sticking, because it's going to stick to your lungs.
Approach in a different way, like using a different technique, like using a microscopy or something? Not like our traditional D10, D50, D90, like I want to go with the microscope.
Well,
Is
if it
that
sticks to your
acceptable?
microscope slide, there's another issue. If it sticks to the microscope lenses, I don't know the nature of the sticking. The other thing that you have to consider for a number-based analysis is that if you want a standard error of 1%, you'll need to look at 10,000 images, 10,000 particles to get that standard error on the mean. So there are some statistical considerations to think through as well if you're going to use a microscope. But yeah, I mean, I would try and investigate the causes of the sticking. Why is it sticking, and eliminate them.
If it is really, you know, that material is hygroscopic, you try to establish the dynamic vapor absorption at what RH, what is the percentage uptake, and try to reduce your humidity at the room whenever you're analyzing, like a RH of the room, control it at a 30% level or 40% level. And it can, like, we usually done couple of measurements in that way. It really works.
Like this is a very rare scenario, like 1 in 100 maybe. Like I'm handling drug substances every day, like on a daily basis. But this is a very rare scenario.
Okay.
Like, is there any guidance from the FDA or something like we can do something like microscopy, like using?
I have a suggestion from a similar application, not an API, but a very sticky, difficult to handle substance that is probably hygroscopic. So assuming it's not static, this customer built a glove box and plumbed with nitrogen. So basically try to remove, basically build a dry box around the system. So that seemed to work well for them. Maybe that can work for you.
I'll try it.
Yeah.
I'll try it. Thank you.
I'd like to follow up on the two questions earlier. I really liked it. Is about, I think Christine's question about some of the differences across softwares and also even within the same vendor that you have a newer, older version of the software, certainly cross-platform, even bigger challenges. And the second is what this question about validation using the reference material versus using actual testing material. I think those two questions, although they are asking different aspects, but I see their underlying theme, which is the rigidity of our framework, currently how to evaluate the data in the context of when something that may not be so rigid, may not be so predictable, or we don't really fully understand. I think that's where some of disconnect happens. Again, I really don't think I appreciate enough of how difficult it is for the generic companies. Until yesterday, I was hearing the version of software change, how difficult it is convincing QA to implement that change. I don't think I really appreciate that challenges or difficulties. With that being said, I was just thinking about proposing two things, maybe for some of the conversations, maybe further discussions. First, I think is related to, or I want to ask everyone on the panel or on the audience, anyone familiar with the Q12, ICH Q12? Raise your hand if you know what is Q12, which is established conditions. And just by knowing that in the room, we don't have many people raising hands, I think there is kind of some disconnect. Established condition is to try to encourage the flexibility, especially in post-approval setting, how an applicant can change their control strategies based on what is they understood prior to approval. So basically, if you show your understanding, what can be changed, what's the impact of change, therefore that change is built into the approval decision. So therefore, it's not locked into specific parameters. So I want to propose, thinking along that line, is there something we can work towards for particle size measurement, which itself, is very hard to define how we want to log. I think the moment we start logging in, they start to kind of really fall apart. So anything along the line of using established conditions, not exactly, because again, established condition is mostly for general analytical procedures, not necessarily suitable for particle size measurement. But I think the concept or philosophy perhaps applies, which is how do we define known risk and how do we manage the risk, and to allow some flexibility without fully being restrictive in what is acceptable. And second, maybe proposal or even thinking about want to, as a panel and everyone to consider is, I heard about a lot of questions about common kind of pain points, really sticky pain points. Can we have the expert, which are a lot of them in here today, we can work towards maybe a question and answer type of publications, something that we can get consensus on, and we put it into public domain that weighs the weight of technical expertise from the vendors, and not just one vendor. We want everyone almost consensus that this is what we all agree on. What should be validated? What should be, can we use number-based for dynamic light scattering? This is still a debate that doesn't matter how many times happen, it will continue to happen. And then this question about using the reference material, if I validated my method, I need to validate it using my reference material for my sample preparation. This is sadly is occurring too often that I think we need some kind of the consensus that this doesn't need to be. I know we all shake our hand. No, this shouldn't be this. So simple, but it is so simple that we don't have a consensus that's publicly available. So maybe something to working towards, Q&A, something we can agree upon that we can just put it out there and then settle. And there's still something that we don't understand. Therefore, we can continue to recognize this is something we don't have a solution that we can working towards. I don't know, I didn't pose a question, but I want to hear from what you thought. And maybe more for the later afternoon discussions. If anyone want to respond.
Yeah,
please.
regarding the established condition, I think it would be a good idea that we at least create a single background for all of the industry, how we want to do it for particle size. And for the generic is a really pain point, because I think what Bernan presented regarding the question is there was a relatively high number of questions to tighten the PSD specification. As I said, we have 3 samples at that stage, and to tighten the specification in this stage, we do tighten it because we need to get to approval, but that's a pain point after that. In the QC lab, there are batches which is out of spec and a big issue there. So what we can do is, if there will be an accelerated process for established condition for particle size after the approval, that would be a good, way out, because we can have a wider specification for the submission agreed on. It's not, huge wide. And then we can work together to tighten the specification when we have enough samples using established condition approach. That would be a very good way out.
Yeah, and also I would like to touch upon the fact that there is always the risk of FDA asking more questions. You provide your established conditions, because it's, when you say established, it's theoretical, right? You don't have the batches, you don't have the data to support it, but you're kind of putting out a picture there and telling them, hey, this is what I think is gonna happen in the future, and this is my design, and I would like to have a certain change happening post-approval. Are you comfortable with it, right? Now, nobody has the data to really say, yeah, we can go forward with this. So would this actually putting in a proposed established condition, would it delay approvals? Because the generic industry, runs on a margin, right? And time is everything. So my ask is, would FDA, be willing to not only reduce the filing category, not, from a PAS to a CB30, for example, but also, buy in the idea. Okay, we have this established condition. This is what you'll want to go with, go forward. Give them the benefit of doubt, and then say, okay, if you do this as a reward, yeah, you can go with the CB30, right? And of course, because the CB30, the granting is happening in 30 days, but the review may take a longer time, and FDA still has the opportunity to ask questions. So if this is something that can be, agreed upon, or, FDA can publish some sort of a guidance or some thoughts on this, this would really help the industry to, use ICH Q12.
Clarify, when I was mentioning the ICH Q12 established conditions, I'm more looking towards the vendors. I know that's not something that vendors have at all considered, established conditions, but basically the concept is, what change do we consider is creating an impact on the measurement? If we change the software version, we introduce a new feature, we can define that feature change, what impact it does on the measured output. And then this would alleviating the burden from the actual generic, or it doesn't matter, from pharmaceutical companies from justifying that change. So it's a little bit shifting the burden from in generic companies into more of a manufacturer of the equipment. Again, software changes, things that may introduce. It's a great feature, but we cannot use it because our kind of process locked into the previous version. But what about, we introduce this new feature, but it doesn't impact, and we can show how it doesn't impact. So, it's more of, shifting the risk to where the risk change occurs. If makes sense?
So
Yeah, I think we discussed this yesterday, between differences in vendor instruments, differences within the same vendor's instruments, when there's upgradation, master size of 2000 to 3000, you have these issues. And we also discussed about how validation requirements change. And then when you're revalidating and submitting, you're typically stuck with either a major or a prior approval supplement, which takes a long time. And then you cannot implement this new method until that is approved. So yeah, there are challenges. And of course, yesterday our ask as well
to the vendors was that could they come up with some sort of a bridging study between the different instruments and come up with some sort of what is acceptable, what bridges the differences and what parameters can be implemented. That way, if there is an agreement from both the vendors and FDA, then probably it makes it easier for industry, right, to adopt, to move between instruments, upgraded versions of the instruments without doing a lot of validations. And probably we could even ask FDA for a lower filing category.
Great. So, yeah, so.
I
in
just
the-
may suggest, again, sorry, just suggestion for the vendors that any change in feature or upgrade should be followed with two comments regarding that upgrade affecting the measurement and affecting that CFR Part 11. That would be very good for us to, challenge the FDA that, okay, this upgrade doesn't need any revalidation or rechecking of our method validation or CFR Part 11 validation.
So in the interest of time, we'll have to table this discussion, but it is great for the waypoint. I want to thank everybody who participated, all of our speakers and panelists today, so we can thank those guys. And we're going to pass it off to Yan Wang to give the closing remarks for the virtual sessions for the day. Thank you very much.
Hello, everyone. Before we officially close the virtual portion of today's workshop, first of all, I'd like to thank all the speakers, panelists yesterday, today, and all the audience in person or virtual. I'd take a moment to reflect on what we have accomplished together over the past 2 days so far. We began by exploring dynamic light scattering on day 1, and then we turned our attention to laser diffraction today. We heard not only the theoretical foundations, but also the practical realities of applying those technologies, how to design studies that are robust, reproducible, repeatable, how to validate methods effectively, and how to avoid common pitfalls that could potentially weaken your regulatory submission. We also gained valuable insights from industry and the vendor colleagues. We had candid discussions about how to align measurement outcomes with regulatory expectations. It gives us a better picture of both the hurdles that remain and the opportunities ahead. Advanced data analysis, emerging technologies, and creative approaches, all those can help us bridge the gaps. But to me, what stood out the most is the recognition that true progress will continue to require collaboration across regulators, industry, vendor, technology producers, and the overall scientific community. This conversation is really reinforced an important point I think has been talked through the whole workshop. Particle size analysis is not just about running an instrument. It's about building confidence in the method, in our data, and ultimately in the quality of the product we aim to bring to patients. So this workshop has obviously not solved all the problems, and it was not meant to. Instead, it has given us a better picture of where the challenges lie and where our collective energy can make the greatest difference moving forward. So to those who you join us virtually, thank you for your attention. Your participation has been critical to this workshop. Even though you weren't able to join us in person, to really see the wonderful demonstrations and the very interactive small group discussion, your questions are recorded. Your engagement is greatly appreciated. They have shaped our dialogue, and we will continue keep those questions in mind moving forward. We hope you will take the insights you gained from this workshop back to your organization to continue the conversations there. That's how the impact of this workshop will extend well beyond these 2 days. With that, I will conclude the virtual portion of this workshop. And for our in-person attendees, we'll take the lunch break before moving into the final sessions in the afternoon. So please take this time to recharge and prepare for the more interactive work ahead. And if I could plug in an advertisement, we have a FDA-sponsored workshop, 2-day food workshop in October. I think on October 7th, 8th. That's traditionally the SBIA workshop. So lots of regulatory important issues will be discussed there. I encourage everybody, if you can, register, and you will gain very valuable insights on many sticky regulatory questions. With that, I'll close the virtual portion and